Okay, thank you. I must also thank the organizers of SOSA for affording me the opportunity to present my research, as I'm only a PhD student, and being able to present alongside such experts is quite an honor. Now, continuing on the topic of prostate cancer, I will be looking at the potential of Annona muricata bark extract and Annonnacin in improving prostate cancer outcomes via in vitro evaluations. This project is supervised by Professor Rupika Delgoda and Prof. Helen Asimoto. Now let's look at prostate cancer. Prostate cancer occurs when there's an uncontrolled proliferation of cells in the prostate gland. It is the second most commonly occurring cancer in men worldwide, while in Jamaica, it's the leading cause of cancer-related deaths. Common treatment options of prostate cancer include surgery, radiation, and chemotherapy. These treatment options are sometimes they're expensive, they come with significant side effects, and they're often not readily accessible. And as a result, patients turn to the use of natural remedies. A fairly recent survey conducted in Jamaica, published in 2017, highlighted the high prevalence of use of medicinal plants among cancer patients. This was part of my initial work for my PhD to garner information about the use of medicinal plants by cancer patients. And what we discovered was that 80% of cancer patients in general use medicinal plants therapeutically. Specifically, in the prostate cancer patient population, the prevalence of use was 72.9%. We also identified the most commonly used plant as Annona muricata, used by 52% of prostate cancer patients. This formed the basis of the current study. So Annona muricata is a slender tree with glossy oblong leaves cultivated primarily for its fruit. However, the plant has numerous medicinal uses in which the leaves and the bark are also of importance. In Jamaica, the leaves are used traditionally to treat nervous problems, while the bark of the tree is the most understudied plant part, which leads to our study objectives. So, following an extensive investigation of multiple types of extracts, in this study, our objectives were to investigate the cytotoxicity and underlying mechanisms of cell death in prostate cancer cells induced by the ethyl acetate extract of Annona muricata bark and Annonnacin, and also to evaluate the anti-metastatic potential of the EAB extract and Annonnacin. The ethyl acetate extract of Annona muricata bark is hereinafter referred to as the EAB extract. So in order to do this, first the extract was standardized for Annonnacin using HPLC-MS. The chromatogram can be seen on the right. The extract was then used along with Annonnacin to conduct biochemical investigations of apoptotic markers in vitro. These include the generation of reactive oxygen species, the impact on mitochondrial membrane potential, annexin V assay, caspase 3,7 activity, acridine orange, ethidium bromide staining, as well as the chorioallantoid membrane assay and vascular endothelial growth factor. Using the MTT assay, the impact of the extract and Annonnacin on cell viability was assessed against two prostate cancer cell lines, namely DU145 and PC3, as well as a normal prostate cell line, RWPE1. This impact was then compared to the impact of docetaxel, a standard chemotherapeutic drug. These results are reported as the IC50 values. which is the concentration at which 50% inhibition of cell viability is achieved. And as we can see from these results, both the extracts and annonnacin were able to inhibit the growth of DU145 and PC3 potently, as opposed to its effect on the normal prostate cell line, showing a negligible impact. This is in contrast to docetaxel, which shows potent impact on all 3 cell lines, including the normal prostate cell line. This highlights the selectivity of the extract in affecting only the prostate cancer cell lines. In order to understand how the observed cytotoxicity was achieved, we investigated several apoptotic markers. The first of which was the generation of reactive oxygen species. And what our results show was that neither the extract nor annonnacin were able to increase the percentage of reactive oxygen species generated in the cells when compared to the control, which was untreated cells. In contrast to hydrogen peroxide, which is a widely used apoptotic inducer, hydrogen peroxide was able to increase the ROS generation by 3-fold. We then looked at the impact on the mitochondrial membrane potential, and our results showed that there were no change in the mitochondrial membrane potential when compared to the control, again, of untreated cells and or positive control hydrogen peroxide. We also looked at the impact of caspase 3-7 activation, which are key players in apoptosis. The graph shows that there was no activation of caspase or 3-7 indicated in the cells as detected by the caspase 3-7 green reagent, suggesting a caspase-independent method of cell death. To further confirm the absence of the apoptotic pathway, the human annexin V assay was conducted, and statistical analysis showed that there was no significant levels in the no significant increase in the concentration of annexin V levels that were detected. So since all the apoptotic markers were negative, we applied a double staining using acridine orange and ethidium bromide to qualitatively detect the mode of cell death. The results were viewed under confocal microscopy at 8x10 magnification. So image A shows cells in the control group representing the untreated cells. They fluoresce green, signaling the uptake of the acridine orange stain, in contrast to images B and C, which represents our extract and annonnacin respectively. You have a lighter green fluorescence with cells appearing to fluoresce red, signaling the uptake of the ethidium bromide stain through damaged cell membranes, suggesting a necrotic pathway of cell death. And there were also no nuclear condensation or membrane blebbing observed, as those are characteristics of apoptosis. So we concluded a necrotic form of cell death. So since most chemotherapy drugs are administered as adjuvant therapies, we decided to investigate the impact of the EAB co-administered with docetaxel in vitro by the MTT assay. Interestingly, our results indicate that the cytotoxic impact was greatly improved, as we can see here from the graph that the IC50 of docetaxel was reduced by 50%. We hypothesized that a likely synergistic interaction underlies this improvement in the impact of docetaxel in the presence of the extract on the prostate cancer cells. However, that hypothesis is left to be examined or investigated. Now, owing to the fact that the high mortality rates observed among prostate cancer patients is attributed to metastatic stage disease, we investigated the impact of our extract and annonnacin on angiogenesis, a key stage in cancer metastasis because cancer cells require the formation of new blood vessels to spread. The anti-angiogenic potential was examined via the CAM assay using fertile chicken eggs, and the results were reported as the angiogenic index, which is the number of capillaries counted at the end of the experiment. And from our results, we can see where the extract and annonnacin inhibited angiogenesis based on the angiogenic index that we obtained in comparison to the negative control, which was the eggs treated with L-arginine. Annonnacin specifically showed significant reduction in the angiogenic index, similar to what is observed for our positive control tinzaparin. To further validate the impact on angiogenesis, we conducted a vascular endothelial growth factor analysis, and our results showed that or extract at various concentrations and annonnacin were able to significantly reduce the levels of vascular endothelial growth factor in the cells when compared to the untreated cells that were used as a control. So if we're able to inhibit VEGF and angiogenesis, we can stop the supply of nutrients and oxygen to the cells and prevent migration of the cells from the primary tumor and halt the metastasis process. So in conclusion, both the extract and annonnacin are able to reduce the viability of prostate cancer cells via a necrotic cell death mechanism, and they potently impact the cancer cells in stark contrast to their impacts on normal cells, highlighting their selectivity. Both the extract and annonnacin are also able to inhibit metastasis through inhibition of angiogenesis and the vascular endothelial growth factor. Furthermore, our extract is able to improve the efficacy of a known chemotherapeutic drug, docetaxel, when used in combination to enhance cell death. So taken together, these results support the need for further in vivo investigations into the use of Annona muricata, hence presenting an attractive candidate for the development of new pharmaceuticals targeting prostate cancer in aim of improving patient prognosis. This research was supported by grant funding from the National Commission of Science and Technology, Jamaica, and the National Research Fund, South Africa, as well as grants from the U.S. National Cancer Institute, CARFA, the UADF, the Office of Graduate Studies and Research, and I would like to acknowledge my supervisors, Prof. Delgoda and Prof. Asimoto, for their guidance and contribution to this research, as well as the colleagues of the pharmacology department at the University of the Free State, South Africa, where I conducted most of these experiments. And I would also like to thank my colleagues at the Natural Products Institute, UIMONO. Thank you.
You're muted, Dr. Fletcher.
Sorry. Thank you, Ms. Foster. Are there any questions for Ms. Foster?
I do see.
Go ahead, please, go ahead.
So in the chat, I do see the first question in the chat asks, how did you determine the cell viability?
Okay, so the cell viability was determined by the MTTSA.
Okay. Okay. Oh, wait, they're coming in. Sorry, hold on. They just keep scrolling up in the chat because they're coming in so quickly. It's wonderful. All right. The second question is asking how do you choose the EAB extract concentration to test its synergy with the drug where it improves efficacy? Oh, actually, yes. And then I skipped a question, but I'll go back to that one afterwards. So repeating for you, how did you choose the EAB extract concentration to test its synergy with the drug where it improved efficacy?
Okay, so it was screened, the EAB extract was screened initially in combination with varying concentrations of the docetaxel. So we varied the concentrations of the EAB extract initially, and the concentration we gave us the greatest impact. We selected that concentration, kept it constant, which was 100 micrograms per ml, by the way, kept it constant and varied the concentration of docetaxel.
Perfect. And another question came in only because the feed was kind of choppy for Dr. Lu, but the age guidelines for prostate cancer, if you could just repeat that for her because it was a little bit choppy for her on her end with the Zoom.
Could you repeat, please?
Oh, sorry.
I didn't hear that one.
The question was the age for screening. She was unable to hear at that time, so I just wondered if you could if you could repeat the age for screening for prostate cancer in Jamaica with men.
Dr. Webster, I think that was from the previous presentation.
Yes.
Oh, yes. Okay. I was able to answer in the chat too, so that's great. Okay, perfect. And then below that, what is the ethnic background of the tested cell lines?
So these are primarily Caucasian cell lines obtained from ATCC. We would love to have some African American cell lines to test, but we were unable to get them, so we had to work with what we have, and these were Caucasian cell lines.
Perfect. And we have some kudos in here as well. Congrats to all the Natch Medicine researchers. Just throwing that up there. Another question, with the high prevalence of use of teas in Jamaica, what is your plan for this? Will you continue to screen the tea or move on with the Annonnacin or both?
So, well, we've actually screened some aqueous extracts previously. So what we realized though from our results is that the aqueous extracts, they're effective at higher concentrations. So concentrations like 300 micrograms per ml or 250 micrograms per ml. So essentially we would move on with the compound, the Annonnacin and this extract, because it was the most potent of all the extracts that we tested.
Perfect.
Ms. Foster, I have a follow-up question. So I see there's a high prevalence of use of the tea, the SOURSOP tea or extract. Do they show any benefit of the use in cancer patients?
Well, anecdotal evidence suggests that patients use these plants because of perceived efficacy.
Okay.
So, yes.
So it's perceived efficacy, but there hasn't been a controlled study?
Right. There are no clinical studies to date in Jamaica using the Annona, Muricata and prostate cancer patients, but we hope to include that in some of our future studies to see what the impact would be in vivo.
Thank you.
Awesome. And back to the chat. So how did you determine the number of new capillaries? Can you please explain how you use the chick membrane?
Okay, so for the CAM assay, the fertile chicken eggs were obtained, they were incubated, and after which the window was opened to see the blood vessels growing. Then we soaked some discs in our samples, and then these discs were then placed on the capillaries in the chicken egg. It was resealed and incubated for another 4 days, after which the egg was opened on the 11th day after incubation, and the capillaries were counted microscopically.
Perfect. Thank you. Another question for the speaker. Great work. So kudos out there to you. So you mentioned this was the bark. Are there any compounds also in the leaves? If so, do you know the levels?
Yes, the leaves does possess the same acetogenins as seen in the bark, but the quantity is usually sometimes higher in the leaves than the bark, but the leaf is the more studied part of the plant. So the levels are usually higher in the leaves, but in the fruits, the compounds tend to be a little different in the fruits.
So they're
with lower levels. So the most potent part really would be the leaves, but the activity is specific to different cancer types.
So follow-up question to that. So, Jamaica is such a natural country. People like to just walk and pick fruit, Is it a possibility? I mean, I know you say the levels are low in the fruit, but let's say someone has this fruit in their diet on a daily basis. Do you think that that there is protection from that?
So it's possible, but we would have to look at chemo prevention studies in order to show that scientifically, and that's not yet done.
Perfect, thank you. Scrolling through the chat, does anyone have any other questions? Feel free to raise your hand in the participant list. Oh, great, another question came in. Kudos to the speaker. It says, great results. Do you think these data would travel? What am I saying? I must be tired. Do you think data would transfer to other cancers?
So, well, for the bark extract is so selectivity towards prostate cancer cells. Because we've screened also the MCF7, which is the breast cancer cell line, and we screened also against HT29, which is a colon cancer cell line, and we saw the most potent impact on prostate cancer cell lines. Ooh. So there's some sort of selectivity from the bark extract.
That's interesting. So nothing for colon, nothing for breast, but for prostate. And that's interesting because when you have mutations, for example, in the BRCA gene, you are at risk for breast and prostate. So it's curious to think that it don't serve any purpose. Like there's no correlation. Yeah. Dr. Lowe, did you raise your hand? Do you have a question? You might be muted. Oh, okay, great. All right. Well, it seems that we are, oh, another one just came in. So what are the adverse effects from the extract?
So from the extract, thinking about adverse effects, we actually looked at screening against a common drug metabolizing enzyme, that's the cytochrome P450-3A4, which is a common drug metabolizing enzyme, and we saw there was moderate to weak inhibition of that enzyme. Hence, the adverse effects are negligible, looking from that angle. And as our patients who use them also, they report no side effects and no adverse effects.
That's great news. Great news. All right. Well, it looks like the chat is all out of questions now. Dr. Fletcher, back to you.
Well, thank you. Thank you, Ms. Foster, for your presentation. Great presentation.
Thank you very much.